Gene therapy. What are gene therapy and cell therapy?
Gene therapy makes use of genes in the treatment or prevention of genetic defects including mutation, and some acquired diseases. It may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. There are two types of gene therapy, somatic and germline. Cell therapy utilizes human cells transferred into a patient to replace or repair damaged ones. Examples of this include blood transfusion, bone marrow transplants or hematopoietic stem cell transplantation and mesenchymal stem cell therapies.
How does gene therapy work?
Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Genes may be introduced, removed, or changed in the cells of patients receiving gene therapy. This mostly experimental technique changes how the cell produces proteins. As mentioned earlier, there are two types of gene therapy, somatic and germline. Somatic gene therapy involves the transfer of a section of DNA to any cell of the body that doesn't produce sperm or eggs, while germline is the transfer of a section of DNA to cells that produce eggs or sperm. The strategies being used in gene therapy include gene addition, gene correction, gene silencing, reprogramming, and cell elimination. Gene addition is a strategy that involves addition of functional copies of a gene to address the underlying genetic cause. Gene correction allows provision of the tools for both repairing and mutating DNA, for discovering gene functions and for engineering new genetic variants. Gene silencing is the regulation of gene expression in a cell to prevent the expression of a certain gene. Gene silencing can occur during either transcription or translation and is often used in research. Reprogramming is the process of changing one cell fate to another. Cell elimination removes the cell completely by delivering suicide agents into the target cell.
What are the uses of gene therapy?
Gene therapy can be used in replacing missing or defective genes, delivering genes that speed up the destruction of cancer cells, supplying genes that cause cancer cells to revert back to normal cells, delivering bacterial or viral genes as a form of vaccination, providing genes that promote or impede the growth of new tissue,